Hello, everybody. Hope you're doing well. I'm Aisha. I'm a third year medical student. And in today's video, we are going to talk about the introduction and history of microbiology. The reference to this chapter is from Essentials of Microbiology by Apurva Shastri, and I have split this into two sections. So, firstly, I'm going to give a brief overview of what microbiology is, what are microorganisms, and how will we deal microbiology throughout the year, how the chapters are going to be split up, and so on. And after that is the main section, which is the history of different scientists and how they have contributed to microbiology. And this is an important section because you need to remember the different discoveries of scientists for your general knowledge, and also if you're giving any exams, then it will help in the MCQs. And if you're giving university exams, then some important questions might be they might ask you to write about Louis Pasteur and Koch's postulates. So these are some important questions that can be asked. So to make it easy to remember, I have made some mnemonics, and do not worry, the mnemonics are not something that you will have to memorize. They are just going to be some smart tricks and tips to remember the discoveries. So let us begin. Firstly, let us start with what microbiology is. So it is the medical microbiology. It is a branch of medicine, and it deals with the study of microorganisms. That's pretty simple, right? And what are microorganisms? This is pretty basic, but I'm just gonna revise it anyway. So microorganisms are divided into prokaryotes and eukaryotes. Prokaryotes are the one that have a more diffuse nucleus, and eukaryotes are the one that have a well-defined nucleus. What I would like you to do is to just revise the table that is present in the textbook, or just search it up and then just read about the differences between prokaryotes and eukaryotes before we move on further. Or if you remember it, it's okay. Let us just move on. So, prokaryotes are your bacteria, and eukaryotes are fungi and parasites. And we have viruses that are neither prokaryotes and nor they are eukaryotes. They are different because they lack the characteristics of living organisms, except that they have the ability to replicate. So these are microorganisms, and when we study this is microbiology. That's pretty simple. So this is a broad definition. When we talk more specifically, that's when we talk about our clinical microbiology. Now, what is clinical? Anything that we apply is clinical in your daily practice. So, when we are diagnosing a patient that's coming with infectious diseases, or when we are treating them, or we are taking measures to prevent infectious diseases, it comes under clinical microbiology. So, clinical microbiology is further split into these sections. And this is how we are going to study our microbiology. This is the structure that is followed. So if you understand the structure, it will just be, easier for you to study and also understand in what way are we moving throughout. Okay, so let's understand what general microbiology means. So, general microbiology will talk about, let's say, we're talking about bacteria. So, the classification of bacteria, the lab diagnosis of bacteria, and if we are having bacterial infections, how are we treating them? How does bacteria cause infections? So, the pathogenesis, all of this comes under general microbiology. So, your bacteria, parasite, and all of that. And your microscopies, what different microscopes do we use? So that is all general. Next is your immunology. Immunology is a study of immune system. But apart from that, how does an infectious disease affect your immune system? And how do we fight back? And what are some immunological tests that we use for lab diagnosis? Let's say a bacteria affects your body. It gives the antigens, right? And then your body produces antibodies. Now, how are we detecting these antibodies or these antigens? There are different tests like ELISA, there's tests like immunofluorescence assay. So all of these we'll study in immunology. Next is your HIC, which is hospital infection control. So as the name says, control. So this is coming under your prevention. And also in this chapter, we'll study about different ways in which we'll deal with your biomedical waste, because it can cause infectious diseases, right? And your hospital acquired infections, like what are some infections that you can acquire while being in a hospital, and how do we prevent those? How do we control those in a hospital? What are some sterilization and disinfection techniques that we have? Why do we wash our hands? What's the technique of washing hands, your hand hygiene? How do we keep hospitals clean? All of that we'll study in this chapter. And then systemic. This has recently been introduced, or previously, micro was taught in an organism way, like all the bacteria together, all the viruses together. But now it has been shifted to this pattern. So instead of all bacteria together, they have split it into, let's say, CVS. So what are some bacteria that cause CVS infections? What are parasites that cause CVS infections? Fungi that cause CVS infections, and so on. So these are all the chapters that we'll deal with in systemic, which is blood, cardiovascular system, gastrointestinal system, hepatobiliary system, skin and soft tissue infections, central nervous system, respiratory system, urinary tract infections, genital tract infections, your eye and ear, and other systems. So in others, and also there will be a chapter called zoonotic infections, and so on. Okay, so this is just a brief overview of microbiology. If you want to give a reading, then do give a reading, and also read about the differences between prokaryotes and eukaryotes. Now, let us move on to our next section, which is important, which is the history of microbiology and the scientists that have contributed to the microbiology today. Now, so starting with Anton von Leeuwenhoek, we have heard about his name a million times. So, Anton von Leeuwenhoek was the first guy who saw bacteria because he discovered a microscope. So, it is in his name. LE stands for lens. So, he discovered a single lens microscope, and whatever he saw in it, he named it little animalcules. Okay, so this is how we'll remember it. Leeuwenhoek, L is for your lens. So, single lens microscope. First person to look at bacteria in a microscope, and he called them little animalcules. Next scientist is Edward Jenner. Again, we've heard him a million times because he made the first vaccine, which was a smallpox vaccine. So there were some children that had smallpox vaccine, and then he used the cowpox virus to immunize those children. So cowpox was called variola vaccinae, and thus the word vaccine comes. Okay, so Edward Jenner discovered the first vaccine. Leeuwenhoek saw bacteria for the first time, and he discovered the single lens microscope. Moving on, Louis Pasteur. He is very important, and everything about him is in his name. Okay. So, Louis Pasteur, let's begin with Pasteur. So, with Pasteur, he did pasteurization. So, what is pasteurization? Pasteurization is done of milk. It's when we heat the milk to kill the microorganisms, in easy words. This we'll study both in microbiology and in third year in SPM, because pasteurization is important in a community, the pasteurization of milk. And then in SPM, you'll also read about how if the milk is like completely pasteurized or not, how to check those things, and so on. So let us leave this for now. I just want um, two things from you. The first thing is, whenever you read a word, understand the word. What are we talking about? Second thing, especially in microbiology, whenever you come across any microorganism, please understand what kind of a microorganism it is. Don't just read the name. Please know if it is a bacteria, if it's a virus, if it's a parasite, and fungi. Because if you keep looking at what it is, you will remember it by the end, and your memory is going to be strong. Okay, continuing. So, Pasteur is for pasteurization, and also Pasteur Institute Paris is named after him. Okay, next, with his name, Louis, so L is for liquid media. Now, what is liquid media? You know, your microorganisms can be grown in like cultures, right? So they can be solid or they can be liquid. So he was like, oh, you know what? I can like use liquid media and I can grow my microorganisms in that liquid media. And he was like, wow, I can do that. And next, with the S, is sterilization techniques. Remember how we were talking about hospital infection control, and then I told you that there are different techniques for sterilization and disinfection. Let's say a doctor is going to perform a surgery, they have to use scalpels and different instruments. Before that, there's a process called sterilization. So, who came up with this? This guy came up with some sterilization techniques like steam sterilizer, hot air ovens, autoclave. Basically, you put your instruments in like a hot oven, for example, and the heat kills the microorganisms. And we'll study about this in detail in HIC, so don't worry. So what do we have to remember till now is that Pasteur, he discovered pasteurization. Pasteur Institute Paris, L is for liquid media, and S is for sterilization techniques like steam sterilizer, hot air oven, and autoclave. Moving on, he is a very great person because he is the father of microbiology. He has done so many things. Like, look at the amount of things this person has done. So, father is our mnemonic. So this is our mnemonic, which is father. Let's begin with F. So F stands for fermentation principles. Basically, the people before वो लोग believe करते थे कि when the fermentation process takes place, it just takes place spontaneously. But he was like, no, 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 it doesn't take place spontaneously. He said that there are microorganisms like yeast which are responsible for the fermentation. So he gave the fermentation principles. Next, for A, we have the anthrax vaccine. So he actually gave the vaccine for three organisms, which are cholera, anthrax, and rabies. Like I told you guys before, whenever we come across a microorganism, we'll see what type of a microorganism is it. Is this a bacteria? Is this a virus, parasite, or fungus? Okay, so let us look. He gave three microorganisms, right? So first, let's begin with cholera. So cholera is caused by Vibrio cholerae, and it's a bacteria. We know Vibrio are like comma-shaped bacteria, right? So these are gram-negative bacilli. Now, obviously, this is a little too overwhelming, but I'm just gonna discuss it very briefly here, okay? Our bacteria are divided into gram positive, okay, and gram negative. What does gram mean? It is based on gram staining, which we'll come to when we are discussing our general microbiology. But I just want you to know that एक stain होता है which is a gram stain. When you stain your bacteria, some bacteria give a color which is purple color. If they give a purple color, they're called positive. If they give a negative, which is if they're like pink in color, it's called negative, and it's called gram negative bacteria. तो gram stain है. Two types of bacteria बन गए. Positive are the ones that turn purple. Negative are the ones that remain pink. Normally they are pink. If they take up the stain, they become purple. Okay, so that's why they're called gram positive. Now, Vibrio cholerae are gram negative. Now, further, your gram positive or your gram negative will be divided into bacilli and cocci. We have read so many shapes, right? Like comma shaped, rod shaped, cocci shaped. But when we are broadly classifying, we only give two shapes. One is bacilli and one is cocci. Bacilli means rod shape, cocci means like spherical shape. Okay, so because these commas kind of look like rods, we'll call them bacilli. So this is not difficult to remember, right? They are bacilli. This is the difficult part to remember. Um when we are talking about bacteria, I'll talk about it in detail. But again, I just want you to know, I'll write it here, that bacteria are positive and negative and gram, okay, gram positive, gram negative, and these can be bacilli or cocci, and gram negative also can be bacilli or cocci. Now, in this, to remember gram positive, gram positive bacilli, we have a mnemonic which is A, B, C, D, L, M, N. Okay. For gram positive cocci, also we have a mnemonic which is very easy. All gram positive cocci having coccus in their name. Okay. Like streptococcus, staphylococcus. So whoever has coccus in their name, ek to see they're having coccus, that means they're positive. Okay, like they're telling ke, oh, I am positive because look, my name itself says I'm a very positive person, and I like always think about having the classification in my name. So he's a very positive guy. So for this we have mnemonics. Okay, this looks really messy. I'm just gonna move all of this. Okay, so the caucus guy is telling clearly that to remember me, it's very easy. Okay, so we have a mnemonic for this, like I said, gram positive. So positive people have mnemonics for them, they are very positive. But for gram negative, we only have a mnemonic for cocci. Whatever organisms that don't fall under this mnemonic and this mnemonic will automatically be gram negative bacilli. Okay, so I know this is very overwhelming, but I just want you to keep this in your mind for now. Okay, moving on. Like I said, gram negative don't have any mnemonic. Vibrio doesn't have any mnemonic. He'll just stay in gram negative bacilli. In Vibrio, it already looks like a bacillus. Next one, we said CAR. He gave a vaccine. Okay, let's not get out of context. We are talking about Louis Pasteur. He gave a vaccine for these three microorganisms. We've talked about cholera. Cholera is caused by Vibrio cholerae. Vibrio is comma shaped. It kind of looks like a bacillus. So it's a bacilli, it's a bacteria, it's a gram negative bacilli. Next one is anthrax. Anthrax is caused by Bacillus anthracis. Now the name Bacillus tells us it's a bacilli. Now, when I was telling you just right now that our bacteria are positive and negative. Positive are bacilli and cocci. These are positive people. They have some mnemonic that I have ready. So now I'm going to talk about gram positive bacilli mnemonic. And for anthracis, the name already says that it's a bacillus. Okay. So bacillus anthracis, it is a gram positive bacilli. Let's talk about the mnemonic for all of the gram positive bacilli. Okay. So here A, B, C, D, L, M, N. Okay. So A is for actinomyces, B is for bacillus. So bacillus, there are two types, serous and anthracis. We're talking about anthracis that causes anthrax. That's not difficult, right? You don't have to memorize this right now. I'm just reading it once because the more repetitions, the more it's going to be in your mind. Okay. So A, B, C, D, L, M, N. A is actinomyces, B is bacillus. It can be serous or anthracis. We are talking about anthracis. It causes anthrax. Louis Pasteur gave a vaccine for anthrax vaccine. Okay. Next one is C. C is for Clostridium. Clostridium can be perferigenes, Clostridium tetani that causes tetanus, Clostridium botulinum, Clostridium difficile. We are going to talk about all of these when we go to our general microbiology, when we are talking about our bacteria. So do not worry, I'm just giving an overview. So when you look at these names again, you'll think to yourself that, okay, I've heard of these before. Okay. Next is D. D is diphtheroids like Clostridium diphtheria, Corynebacterium diphtheriae, which causes diphtheria. If you have heard of it, it is a respiratory tract infection. So CD. And then LMN. L is Listeria monocytogenes. M is Mycobacterium leprae, Mycobacterium tuberculosis. Tuberculosis causes TB. Mycobacterium leprae causes leprosy. Then nocardia. Okay, so these are all your gram-positive bacilli. One last time, I'm just gonna say it again. Your bacteria, on the basis of gram staining, there's a stain called gram stain. They can be positive. Positive look purple. They can be negative. Negative look pink. Further, they are divided into bacilli and cocci. Further, bacilli and cocci. Positive people have a mnemonic for them. The positive bacilli have A, B, C, D, L, M, N, and the cocci have coccus in their name. They are telling you positively that they are coccus, like streptococcus and staphylococcus. Okay. Next one is rabies. Rabies is caused by a virus, which is rabies virus. Viruses are of two types. Okay, viruses can be DNA viruses or RNA viruses. This is easy, right? So rabies starts with an R and it's an RNA virus. That's pretty simple. Now coming back to our topic, Louis Pasteur, he's the father of microbiology. F was for fermentation principles. He said that fermentation doesn't take place spontaneously, it takes place because of some microorganisms. And then A is for anthrax vaccine, and he gave two more vaccines for cholera and rabies. Okay, and then THER is for germ theory of disease. So this guy, he said that any disease, it is caused by, not any disease, sorry, diseases are caused by germs. Okay, people used to think that let's say किसी को हो गया है cholera, so people used to think, oh, is this spontaneous? But no, he said it happens due to some germs. So he gave the germ theory of disease. Same with fermentation principle. People used to think it's just spontaneous. So he was like, it's not spontaneous, it takes place due to certain microorganisms. Okay, so this is about Louis Pasteur. Okay, one last time, let's just see an overview. Pasteur, pasteurization, Pasteur Institute, Paris. L was for liquid media. S is for sterilization techniques, which is steam sterilizer, hot air oven, and autoclave. This field study in hospital infection control. Next, he was the father of microbiology. He is the father of microbiology. F is for fermentation. A is for anthrax. And two more, cholera and rabies. Cholera and anthrax are bacteria. And cholera is caused by Vibrio cholerae, looks like bacilli and doesn't have a mnemonic, so gram negative. This Bacillus anthracis AB, so it is having a mnemonic, so it is positive, and then the name says that it's a bacillus. Okay, next one, rabies. Rabies is R, so RNA virus, and it's caused by rabies virus. Next, THER, germ theory of disease, and it says that disease is caused by microorganisms and it is not spontaneous. Okay, so this is about Louis Pasteur. Let's move on to our next scientist. Okay, our next scientist is Joseph Lister. So in his name again, we can see that SEP. So Joseph, he gave the septic. So antiseptic, he is the father of antiseptic surgery. So he thought ke, oh, I am septic. So he thought ke I need to make something which is against me. Okay, antiseptic surgery. So he's the father of antiseptic surgery. Basically, he used like disinfectants like diluted carbolic acid during surgery to sterilize the instruments, and that's why he is known as the father of antiseptic surgery. No need to like memorize all these details. Just remember that he's the father of antiseptic surgery. Okay, moving on to a very important scientist who is our Robert Koch. Okay, so now he's a very solid guy. He's a very solid, pure guy. See, he's a very important guy because Koch postulates might be asked in your exam. So remember that this guy is a very solid guy. You'll remember, like you'll hear about him everywhere. Okay, so remember his name. He's a solid, pure guy, and he used to like get LPG gas. You know those LPG cylinders, and he used to get like AC for his family. But then one day he had a test and then he hanged himself. So he's a solid and pure guy, and he got scared of this test and he just hanged himself. Okay. So this was a stupid story, but let's read what it means. Okay. So Robert Koch for solid media for culture. See, our Louis Pasteur, this guy gave liquid media. This guy was like, oh, we can use solids also to grow. ठीक है तो he was like let's use solid media for culture of bacteria, but actually this was not his idea. This was this woman's idea. She was the wife of Koch की कोश के असिस्टेंट की वाइफ थी. ठीक है, so she suggested this and then he was like, oh, smart girl, I'll just copy your idea, and he did that. So moving on, next is pure is for pure culture. So, pure culture means a culture which is just having one organism. Let's say okay, we have a culture that is having multiple microorganisms, but then you take one bacteria and then you take a solid culture, let's say, and then you just isolate it. So, this is called a pure culture. So, he was like, you know what? Not only can I grow things in a solid media, but I can also extract some pure cultures. Like I can extract one single organisms. Okay, organism. Sorry. So pure culture is a method to isolate your bacteria. Next, he's a solid pure guy. So with guy you can remember die because it kind of rhymes. But even if you don't remember this, you remember that he had a test and then he hanged himself and then he died. So die. So he gave aniline dye. Okay. So you can remember it in two ways, either by guy or by dye. So he didn't give the dye, but then he discovered that you can use this dye to stain. Okay, like you can do various staining techniques, like I just talked about gram staining, right? Just like that, he was like you can use aniline to stain. Okay, so he did these three things. Apart from that, he gave the Koch postulates. We'll come back to the LPG and AC later, but then let's look at the Koch postulates. So there are total 5 postulates. Actually, there are 4, but then he gave one more later because he realized that, oh, this is also possible later on. So, first one is see, we have 2 organisms, okay? One is very happy because he's not sick, one is having some disease. Now he looked at this sad rat and he was like, what if I isolate this guy's disease lesions? Let's say this rat had some skin lesion. Okay, he was sick, and then Kosh was just sitting here, and then he thought, okay, oh, let me take this and isolate it in a culture because oh, I just discovered that there's something called solid media, and I can isolate this in a culture. And then he was like, oh yes, it is possible. I can isolate microorganisms in cultures. So the first postulate is the microorganism should be constantly associated with lesions of the disease. So he concluded by that let's say this rat is sad because he has some lesion that is caused by some microorganism. Now, obviously, he didn't know because at that time they didn't know that, oh, the microorganism will be present in this lesion. He just randomly isolated this lesion and then he saw it in a culture. And then he realized that that microorganism that caused this disease is present in that culture. So the first postulate is, like in fancy words, it is the microorganism should be constantly associated with the lesions of the disease. That means if someone has a lesion and you isolate it, that means you should be able to look at the microorganism in that isolated culture. Okay. Next thing is, he was like, I also discovered something. See, we just saw that he was a solid, pure guy. So he also discovered pure culture. That means, let's say he saw a rat who was sad. He took out the microorganism in a solid culture, and then he's like, what if I just take one bacteria from this and then isolate it in a pure culture. Pure मतलब वो पहले से pure था. There was nothing growing on in this agar, let's say. But then he was like, what if I take one bacteria, whatever from this, and then I put it in a pure culture? Will it be possible to grow it? And then he realized that yes, it is possible to grow. So he said that second postulate is it should be possible to isolate the organism in pure culture from the lesions of the disease. Okay, so you are able to isolate it in pure culture. Now, next one is that now, whatever was isolated in this pure culture, if I take that and I put it in a healthy lab animal, he should get the same disease that this previous rat had. Okay. So, the definition for the third postulate will be the same disease must result when the isolated microorganism is inoculated into a suitable laboratory animal, and that was happening, right? So same disease is happening when a healthy animal is inoculated with this microorganism that cause the disease. Now the last postulate is now this also became sad. Now it should be able to re-isolate this in pure culture. Okay, so whatever lesion this guy has now, if we take this again, we should be able to re-isolate this in pure culture. So, revising all of these, first one is that the microorganism should be constantly associated with the lesion of the disease. That means a healthy animal should not have microorganisms that the disease animal is having. So, whenever someone's having a disease, infectious disease, they should have the microorganism when we are looking at the culture. And then it should be possible to isolate the organisms, these microorganisms that are causing the lesions. So it should be possible to isolate these organisms in pure culture from the lesions of the disease. So from lesion of the disease, you should be able to isolate this in pure culture, and then this will grow in pure culture. So from this pure culture, when we are taking the isolated microorganism and when we are inoculating it in a healthy, suitable laboratory animal, the same disease must result. So the third postulate is the same disease must result when the isolated microorganism is inoculated into a suitable laboratory animal. And then the last one is it should be possible to re-isolate the organism in pure culture from the lesion produced in the experimental animal. Okay, we are done with four. Now the fifth one he added was that, let's say this guy is having a disease. Now obviously he'll have the antigens that are causing the disease, right? Antigens. See, microorganisms have antigens. These antigens will come in our body and they'll cause disease. Our body's immune system will produce antibodies to fight against these antigens, right? So these antibodies, all of these will be present in the serum. So when we take the serum of an infected animal, and we should be able to see the antibodies to that causative organism. That makes sense, right? Microorganism has antigens. Our body produces antibodies. So when we are taking the serum of someone who is infected, we should be able to see these antibodies that are against these antigens. So the definition for fifth criteria is antibody to the causative organism should be demonstrable in patient's serum. Okay, so these are the five postulates. Please read the definition once so that you'll remember it too. Now, to these postulates, there are three exceptions, which are our LPG. So I said Robert Koch was a solid pure guy, and he used to get like LPG cylinders for his family and also an AC. Now, LPG stands for lepre, P for palladium, G for gonorrhea. Okay, so lepre is Mycobacterium leprae. If you remember, just now I was talking about gram positive bacilli. See, bacteria were positive, they were negative. Positive were both bacilli and cocci, and they had mnemonics for them. So bacilli ka mnemonic was A, B, C, D, L, M, N, and M was for Mycobacterium, Mycobacterium leprae, and Mycobacterium tuberculosis. So leprae is gram positive bacilli, okay, and this causes leprosy. So leprae, Mycobacterium leprae. Sorry. This is sorted. Mycobacterium leprae is a gram positive bacilli because it has a mnemonic A, B, C, D, L, M, N. M is for Mycobacterium. Mycobacterium can be TB or leprae. Okay, moving on. Next one is Treponema pallidum. Now, I told you that bacteria can be gram positive, they can be negative. What if they don't take any of these stains? What if they take like thoda positive? We can't really classify them, right? So, this is that kind of bacteria. It is a miscellaneous bacteria and it comes under this group called spirochetes. So, just remember it now that Treponema pallidum is a type of spirochete and it's a miscellaneous bacteria. Next one is gonorrhea. Okay, so Neisseria gonorrhoeae. Now, again, I'll go back to when I was talking about bacteria being positive, negative, and then bacilli, cocci. This we are done, right? This we know that this is A, B, C, D, L, M, N, and this is like whoever has coccus in its name comes under this. So let's forget this for now. Let's just talk about bacteria that are negative. And then I said that there are bacilli and cocci. Now, your bacilli don't have any mnemonic, so let's forget them. Our cocci do have a mnemonic. You can remember them. They're so easy. See, they're negative, right? Negative. So they start with an NE, which is Neisseria gonorrhoeae, and and again Neisseria meningitis. Okay, meningitis. So whenever you see Neisseria, it's gram negative and it's a cocci. Easy because I said bacilli do not have a mnemonic. Now it's starting with ni negative Neisseria. So Neisseria are our negative cocci. Okay. So Neisseria gonorrhoeae, it is a gram-negative cocci. Now, why were we talking about these again? These are exceptions to our Koch postulates. Okay, so why are they exceptions? Because LNP, the first two, they cannot be grown in vitro. That means you will not be able to do this step for them. It's just an exception. So that's why we are reading these. And then the last one, gonorrhea, it doesn't have any animal model. That means the third postulate will not be applicable because you'll not be able to isolate it in a healthy animal. So when we are talking about these organisms again in detail, these points will be repeated there again. Okay, and that's why I keep repeating the names because the more repetition it's good. I'm sorry if I'm eating your brains up, but this will be very helpful later on. So one last time, Mycobacterium leprae, A, B, C, D, L, M, N. M is for mycobacteria and it has a mnemonic, so it's positive, and it's bacilli because the positive cocci will have coccus in their name like streptococcus and staphylococcus. Next one is Treponema pallidum. It is a miscellaneous bacteria. It comes under spirochetes. Next one is Neisseria gonorrhoeae. Ne, so it's negative and it's a cocci. Okay, gram negative cocci because bacilli do not have any mnemonic. Gram negative bacilli. Next one is our AC. So we are talking about Robert Koch, that he's a solid pure guy and he used to get LPG cylinders and AC, but he had a test and then he hanged himself and he died. So ANC is for ANC is for anthrax, tubercle, and cholera bacilli. AC but okay, anthrax, tubercle, cholera bacilli. He discovered these bacteria. Okay, so we have already studied about anthrax. Who gave the vaccine for anthrax? Louis Pasteur. So anthrax was Bacillus anthracis. B for Bacillus. Um, and this is a gram-positive bacilli because it has a mnemonic. And then our cholera, we've already talked about cholera as well. So basically, this guy discovered these bacteria, and then Louis Pasteur gave the vaccinations against these diseases. Okay. Um, Moving on, test and hang. So, so there's this thing called testing motility. That basically means you're testing the motility of an organism. See, an organism can be motile or it can be non-motile, right? Motile means it has flagella. Non-motile means it could have some Brownian moments or it could just be stable. So he was like, how can I test the motility? And then he was like, wait, I can give a hanging drop method. Now, what is hanging drop method? This is related to general microbiology. We'll read this in lab diagnosis, but I'll just give an overview, not in detail, but just an overview. Basically, we take, see, we just talked about solid cultures, right? Like let's say we cultured some lesion of this animal. Now we want to know what type of bacteria that is. Obviously, we won't get a dream that, oh, this was Mycobacterium tuberculosis. And we know that, let's say, okay, some organism, let's say A organism is motile, B organism is non-motile. We are suspecting that he either has A or B. So if we isolate it and see that if it is motile or non-motile, we can conclude if the organism is A or B. Okay, so that's why we need this test. Okay, so um, what for this what they do is they take a cover slip, and then from this type of culture they'll take like some itna sa part, okay, and then they'll put this on a cover slip, and then they'll put some petroleum jelly here. Let's see here. Okay, so they'll take like a loop. ऐसे loop होता है, ये अगार का plate है समझो, इस पे ऐसे loop लगाते हैं, and then they'll scrape off something, and then they'll take this small cover slip, they'll put some petroleum jelly on the sides of it, and then they'll place this small microbial suspension into the center of the slip. Now they'll take a slide, okay? Now the slide is a special slide because it has some sort of depression. So ये cover slip के ऊपर ये वाली slide को ऐसे place कर देते हैं. Okay, so turn the concave depression upside down over the cover slip. Then they'll suddenly turn this over. So they'll turn it over in such a way that now the thing that we placed in the middle is hanging in this concavity. It will look something like this. It will be a drop of liquid with specimen hanging upside down from under surface. Now we'll put a microscope on top of this and we'll observe if it's moving, if it's not moving, and then we'll conclude if it's motile or non-motile. So this smart guy, he came up with this technique of hanging drop testing motility. Okay, so he hanged himself and died, and dye was for aniline dye. So once more, I'll just revise everything about this guy. He's a solid pure guy and he got LPG cylinders and AC. But then he had a test one day and then he hanged himself and died, and he gave Koch postulates. So solid was for solid media, pure is for pure culture, guy is for dye, aniline dye, and then LPG are the exceptions to Koch postulates. L is for Mycobacterium leprae, which is a gram-positive bacilli because A, B, C, D, L, M, N. And because it's a mnemonic, we have a positive, and it's bacilli because gram-positive cocci have coccus in their name. Next, Palladium, Treponema pallidum. This is a miscellaneous bacteria. It's a spirochete. Next, Neisseria gonorrhoeae. This is Ne, so it's negative. And then the first two, they cannot be grown in vitro. And then gonorrhea, it does not have any animal model. And then A and C, he discovered anthrax, tubercle, and cholera bacilli. And then as for test, he discovered a method to test the motility of organisms, which is called hanging drop method, in which basically you take a cover slip, you place the organism, and you hang it in the middle of a slide in a concavity. This is a special slide that has a concavity, a cavity inside. Okay. And then you look for it in the microscope. So this is what Robert Koch did. And please revise the Koch postulates. Now let's move on to the next scientist finally, which is our Paul Ehrlich. All of these things are not present in the newer edition of Apurva Shastri, but I've just written them for the sake of it. So again, from his name, we can identify what he has done. So first, Ehrlich. So there's a bacteria called Ehrlichia bacteria, which is named after his name. And then there's this phenomena, which is called Ehrlich phenomena, which talks about the toxin and antitoxin interactions. What is meant by toxin antitoxin here is toxin is your antigen and antitoxin is your antibody. So Ehrlich talked about like antigen and antibody interactions. There are various things that he has done, like I think we will study the detail of it in immunology, and it's not really relevant here. But Ehrlich phenomena refers to that when, let's say, a lab animal is injected with a lot of toxin, it goes in an immune paralysis. Like it's not able to fight back by making antibodies. That's what Ehrlich phenomena is, and he like discussed about these interactions in detail. Moving on, the I in his name is for immunology. We just talked about this, right? Toxin, antitoxin is antigen, antibody, and he did something really great in this. So that's why he got a Nobel Prize for immunology. And then CH is chemotherapy. So he's the father of chemotherapy. Why is he the father of chemotherapy, you may ask? Okay, so this guy loves the word A and S, okay? And he um, discovered an arsenical compound, which is salvarsan. And he said that, oh, this can cure syphilis. So chemotherapy means therapy using chemicals. So he uses arsenic compound salvarsan to treat syphilis, and this was the first time someone had used something, a chemical, to treat a disease. So that's why he is called father of chemotherapy. Okay. And then A is also for acid fast nature of tubercle bacillus. See, we just studied about mycobacterium, right? Mycobacterium tuberculosis. These organisms are something called acid fast, which we will study in general microbiology. So don't you worry. Just like gram staining, we just studied gram staining, right? There's something called acid fast staining. So whichever bacteria react to this acid fast staining, they're called acid fast bacteria. Okay. So he discovered the acid fast nature of tubercle bacillus, which are your Mycobacterium tuberculosis. He discovered that TB, Mycobacterium tuberculosis, are acid fast in nature. That means they give a positive reaction when acid fast stain is added. Okay. And then S is for he discovered the staining of tissues and blood cells. Okay, so again revising about him, from Ehrlich, Ehrlichia bacteria, and Ehrlich phenomenon, which describes the toxin antitoxin interactions. Toxin is antigen, antitoxin is antibody. So I is for immunology. He did something great. So he got a Nobel Prize. And CH is for chemotherapy. So he's the father of chemotherapy. Why? Because some arsenical compound, salvarsan, he was like, oh, it can cure syphilis. So that's why father of chemotherapy. And A is also for acid fast nature of tubercle bacillus, and S is for staining of tissues and blood cells. Moving on to our other scientists. These are just a few names, and then we'll be done with this chapter. So first one is Hans Christian Gram. In his name it's written he gave the gram staining, which will differentiate our bacteria into positive and negative. Moving on, Ernst Ruska, E is for electron microscope. So Ernst Ruska gave the electron microscope. Alexander Fleming, everyone knows. Fleming, penicillin. Okay. Next one is Good Pasture. So he was a good guy. He loved little chicks. Okay. in a non-funny way. So good pasture, he gave viral culture technique in chick embryo. Next one is Barbara McClintock. So she M is for mobile and T is for transposons. So she discovered mobile transposons. Next one are these two scientists, Walter Gilbert and Frederick Sanger, because there are two and DNA has two strands. They discovered DNA sequencing. And next one is our Ignace, I don't know how to pronounce his name, Semmelweis, Ignaz Semmelweis. He gave hand hygiene. This is a very interesting story. I'll link a Dr. Mike video that talks about how he discovered hand hygiene in the description below. Do watch it, it's kind of interesting. Basically, in if you're not gonna watch the video, short story, long story short, is that he saw that women who were giving birth to babies in hospitals were dying because of doctors, because the doctors didn't used to wash their hands when they were delivering the babies. So he discovered that the babies are dying because of poor hand hygiene of doctors. And there was this fever, which is puerperal fever during delivery, that the women were having, and the women and the children both were dying. So this guy was like, we need to wash our hands. So that's it, guys. That was it for the video. Hope you enjoyed it. Hope you remember at least half of it. And please revise this again. And I'm pretty sure that you'll get used to all the bacterial names and the stains and stuff. Thank you so much for watching. Please subscribe and share it with your friends. Bye bye.